Presentation, I'm going to be explaining the structure of DNA and RNA. Now we know DNA is the molecule that holds that genetic information, and RNA is the molecule that's involved in transcription and translation, in actually translating that code into a protein structure. So both very, very important molecules that we need to learn about. So these are our syllabus points. We're going to be looking at the structure of DNA and RNA and the fact that they're polymers of nucleotides. We're going to be looking at how DNA and RNA differ from each other. We're going to understand the structure of DNA, that it's the double helix with two anti-parallel strands of nucleotides linked together by hydrogen bonds and complementary base pairs. And we're going to draw a simple diagram of the structure of single nucleotides of DNA and RNA using circles, pentagons, rectangles to represent those phosphates, pentoses and bases. Okay, so we're just going to start with a molecule of deoxyribose. This is our a monosaccharide. We've learnt about this before, and if you remember, this is very, very similar to ribose. If it were ribose, it would have a hydroxyl group, an OH here on the carbon 2, but it doesn't, so it just has an H, so it's deoxyribose. And we can see the carbons here. We've got our carbons 1, 2, 3, 4, and then there's carbon 5 there. And these are going to be important because it's those carbons that we're going to attach the other bits of the nucleotide to. So here is a nucleotide. You don't need to know anything in this much detail, but it does nicely show how the little bits join together. So you can see we've got our phosphate, and that is attached here to our carbon 5. We've got our nitrogenous base, and that's attached here to carbon 1. So this is a simple nucleotide. You don't need to draw it like that. You just need to draw something simple like this. And this is a basic nucleotide, and we've just got a pentose sugar here. So this would obviously be deoxyribose if it were DNA nucleotide, or it would be ribose if it were an RNA nucleotide. And there are 4 different nucleotides for DNA, and the nitrogenous base varies. So you either have thymine, cytosine, guanine, and adenine. Whereas in RNA, you've got that one, you've got that one, you've got that one, but instead you've got uracil, which I'm going to show you in a minute is very, very similar. So here we go. You can see that we've got, these are the bases, guanine, cytosine, adenine and thymine. And uracil here in RNA is very similar to thymine, but it's just very, very slightly different. And you can also see that guanine and adenine are bigger molecules, and cytosine and thymine are smaller. And this also shows the hydrogen bonds. You can see hydrogen bonds that link these together in the DNA molecule. Okay, so what I'm going to do now is draw a molecule of DNA and show you how you can draw this nice and easily and how you can label it to show the structure of DNA. So we start off with drawing our nucleotides. So let's get that. Here's our nucleotides. And we'll draw another one. A longer one. You remember I said that adenine and guanine are longer than thymine and cytosine, because they're slightly different molecules. Okay, and we're going to join those together to form one of the strands, and we join it to the carbon 3, so the phosphate gets joined to the carbon 3. And I could continue adding more and more nucleotides, but I'm just going to stick to the 2 for this particular diagram. And then we need to draw the other side. Now, it said in the syllabus points that DNA, the strands are anti-parallel, which means that we have to draw the other strand upside down. So let's do that. So we'll put in our next, and I'll put a long one here, and I'll put a shorter one here. Right, and we're going to draw these upside down. And again here, put in the phosphates, And then once you've done that, you can join the phosphate to the carbon 3 as we did before. So there we go, there's our DNA strand. The last thing we need to do is just to join up the bases with hydrogen bonds. So these are weak bonds. I'm going to join them together like this. And guanine is linked with cytosine, that's with 3 hydrogen bonds, and adenine is linked to thymine, and that's 2 hydrogen bonds. If you forget that, it doesn't really matter. So now we're going to actually label this diagram to explain what's going on. Okay, so we've got here our complementary base pairing. It wouldn't quite work out, sorry about that. And we've got here, this is our sugar phosphate backbone. We've also got our hydrogen bonds, which we need to label here. And we can show that this is anti-parallel, which means that the strands go in the opposite direction. I'm just going to rub this out. This is not very nice. And we'll show it here. So this is our complementary base pair. Much better. Right, other little things that we need to mention here. You could label an individual nucleotide. So we can show here that we've got our phosphate. We've got this, which is deoxyribose, which is our sugar. And we've also got this, which is our base, or our nitrogenous base. So that's quite nice just to label one individual nucleotide as well. And this really explains everything in that learning objective. The other thing you could also draw, if you wanted to, is a molecule of RNA, and that would be very, very simple because it would just be one single strand. So if it asks you for RNA, you would just simply draw. There's one. I might put 3 in a row for this one. Put that in. And I'm going to join them together like that. There we go, that's our molecule of RNA, and we know it's RNA. We're going to put uracil as one of our bases there, and I'm going to do adenine here, and I'm going to do cytosine there. And in this case, we would label this sugar as being ribose. There we go. And that's our RNA molecule.